right, welcome to AMP2. So we're going to jump right in today into Chapter 17, which talks about the functional organization of the endocrine system. Chapter 18 will really get into the different glands and what hormones are produced. We have a ton of stuff to memorize. But this chapter is actually a really nice introduction because one of the things that you should think about is AMP1 ended with the nervous system, right? And the nervous system is using neurons and action potentials and neurotransmitters, right? But what it's really trying to do is send messages to the body or control various aspects of the body. So here in AMP2, we're starting with the endocrine system, which is the other major organ system that controls the body. But we're going to see that it does things very differently, right? It's using hormones that are carried through the bloodstream to pass on those messages, right? In general, I would say if you're looking at the two systems, right, the nervous system was really good at rapid, acute, short-term changes, right? You touch a hot pan and that arm retracts, pulls away from that stimulus. What we're going to see here with the endocrine system is that we tend to, not always, but they tend to be a little more long-term, gradual changes that hormones make, right, to make slight adjustments in homeostasis. They tend to have more widespread targets and that sort of thing. What we're going to do actually on Thursday, this will be super fun, is I'm going to ask you guys on Thursday to kind of compare and contrast these two systems, right? They're both trying to maintain homeostasis, right? They have to coordinate cells throughout the body in order to do this. They are both doing that control with chemical messages, but maybe a little bit differently. And they both use the hypothalamus. Remember that weird little section of the brain? I don't know if you can see that, right? Right? The thalamus is that little brown dot, and just below that, hypothalamus, right? It's part of the nervous system, part of the autonomic nervous system, but it's also going to be basically the organ of highest control in the endocrine system. So these two systems share a lot of different features, and on Thursday, you're going to tell me what they share, and then also how they actually end up being really different as well. So look at that. Okay. So, this is table 17.1 out of your textbook, and here we're looking at the different types of cell-to-cell communication, right? Intercellular communication. I'm only writing this up here to remind you, right, if you took medical terminology, hopefully that was inter is between. Intra would be within a cell. Intercellular communication means between different cells. Okay? And so we have four different kinds here. Autocrine, paracrine, synaptic transmission, and then endocrine transmission. Okay, so when we talk about this cellular communication that's called autocrine, auto should be your big clue that this is kind of self-communication. What we're looking at here is that cells are producing a chemical that's going to stimulate cells of the same type. Not necessarily like self, like the same cell, but of the same type. So one of the classic examples is going to be when helper T cells put out chemicals that tell things like cytotoxic T cells and B cells, right? They're all lymphocytes, to go ahead and replicate and become more active to fight infection. Okay, so that would be an example of autocrine cellular communication, right? Those chemicals that white blood cells release are in a group called leukotrienes. Prostaglandins are another set of chemicals that you might hear, where it's really a very, very localized kind of communication. Our next type of communication is paracrine communication. This is when cells are communicating with their neighbors, right? So this is always within a tissue. They put a couple different cell types here to let you know, okay, this is a little bit bigger than autocrine communication, but we're actually communicating localized with neighboring cells of the same tissue. And so these messengers, often called paracrine factors, or sometimes factors, sometimes local hormones, they're being released into the extracellular fluid, right? All these cells are just kind of bathed in extracellular fluid, and that's how these messages are going to get passed along. You'll see this happen with some different growth factors. So there's one, for example, called vascular endothelial growth factor. As soon as you see factor, you should think, okay, this is paracrine communication, or communication taking place within tissues. Another classic example would be histamine. So part of that innate immune response, this inflammatory response, injured cells, or cells at the site of injury, are going to release histamine, and that's going to trigger other local cells to do things like vasodilation. So paracrine communication, or localized tissue communication. Synaptic communication. This is what we saw with the nervous system, right? So we have one neuron releasing a neurotransmitter, a chemical, across a little gap, a synapse, and it's communicating with a neighboring cell. It's very specific, right? It's always influencing what we would call this postsynaptic cell. It doesn't have to be a neuron, it could be a gland or a muscle cell, anything like that. But it's extremely specific into what cell it's communicating with. And then this takes us into our next chapter, I suppose this chapter as well, about hormones. So by definition, when we talk about a hormone, right, they're not really any different than all those chemicals in those first three kinds of communication, but by definition, right, it's going to be a chemical that's being released into the bloodstream. That's super important. It has to get carried by the bloodstream, and it's actually carried to tissues at a distant location, and that's where it's going to have its effect. Okay? And that's what we're going to be looking at for the next couple chapters. So when we get into chapter 18, we are going to be looking at lots of different glands. A gland, just a reminder, glands are always epithelial tissue, right? And they're going to produce a secretion. If it's an endocrine gland, that secretion goes to the bloodstream, as we just discussed. An exocrine gland is going through a duct to a surface, whether that's the surface of the skin or the inside surface, the lumen of the small intestine, right? We're going to deal with hormones here that are going to the blood. Lots of stuff to memorize in our next chapter. Okay, but let's talk about these hormones a little bit before we get into all of those different organs. So hormones can really be broken down into two main categories. I felt like this part of the textbook got like a little bit clunky, maybe a little bit repetitive. And I think the big thing to keep in mind here is that hormones are either lipid soluble or they are water soluble. Now this is when you're going to be happy if you paid attention in those early chapters on chemistry, which no one ever likes. But if you think back to what those words mean on a chemical level, right? If something is lipid soluble, it's going to be nonpolar. Remember, water soluble, water is polar, and polar molecules dissolve in water. So like sodium chloride can get pulled apart. That would be an example of a polar molecule. Any of your lipids, they're nonpolar. They don't have like a plus and minus end, and they don't dissolve well in water. But you know that, right? Oil and water don't mix. So these lipid-soluble molecules are not going to dissolve well in water. This sets up a few different things for us, okay? So, for example, our lipid-soluble molecules, if they don't dissolve well in water, plasma is basically water. And so what we're going to see with the lipid-soluble hormones is that they're very often, I want to say always, they are carried through the bloodstream by binding proteins. So you would very rarely find them free-floating in the bloodstream, right? So here's our lipid-soluble hormones, and they're trying to show you here, this is figure 17.3, if you need a closer look in your book, the little white lipid-soluble hormones bound to these blue proteins. They, of course, then have to come off of that binding protein to actually leave the bloodstream and act on that target cell. So this binding protein is it's a reversible binding, but it's going to help carry particularly lipid-soluble molecules through the bloodstream. The other thing that being bound to this binding protein, we'll also see that some water-soluble proteins can be carried by binding proteins as well. And the purpose here, if you're talking about either of these, is also to increase the half-life of this hormone. So one of the things, I know where I want to write that. One of the things that happens is these hormones are constantly trying to be removed from the bloodstream by a variety of different forces, right? So when they actually bind to their target cell, that takes them out of the bloodstream, right? That uses them up. But there's also enzymes just circulating in the bloodstream that are trying to degrade these hormones. The liver has enzymes that want to break these hormones down, and the kidney, one of our big filters, will also take these hormones out of circulation. So the other advantage of these binding proteins, right, is not just that it can carry them through the blood, but it also gives them a longer half-life. They end up kind of acting as a reservoir of hormones in the bloodstream. So, for example, like this lipid-soluble hormone came off and is acting on its target cell, but there's more of that hormone kind of sitting there in the bloodstream, ready to be used. Okay? So, particularly the lipid-soluble hormones, since they use these binding proteins, they need to use these binding proteins, they tend to have a longer half-life. And, the definition of half-life too is when 50% half of that hormone has been used up. So how long it stays in the bloodstream. So this tends to be longer than the water-soluble molecules. This could be for weeks or months that these hormones will be circulating. The water-soluble molecules, they can circulate without binding proteins, but there's also quite a few of them that use binding proteins, and in that case it really is to increase its half-life. In general, though, we would say the water-soluble molecules have a shorter half-life. They tend to have a rapid onset, short duration of use. Again, in general, they have a shorter half-life. They just don't last as long in circulation. Okay, so, again, lipid-soluble hormones, water-soluble hormones. We're going to look at a couple other features that differ between here as well. And this is one of those things where you have to kind of memorize these features of each, but once you kind of have hormones into these groups, it's going to be a lot easier to understand kind of how they work. So this might also be a good table to look at to see what the examples of these hormones are. So particularly in the lipid-soluble hormones, we're talking about all the steroid hormones. They're cholesterol-based. You'll see these like ring-like structures. But this is like testosterone, estrogen, progesterone. So all those reproductive hormones are the steroid hormones. Aldosterone is also a steroid hormone, and that deals with sodium levels in the body. Another example, this is actually an amino acid derivative, so it's a protein-based hormone, the thyroid hormones. And then we've got some others. The water-soluble hormones made out of proteins, peptides, pretty much everything else. So if you remember that the steroids and things like thyroid hormone are lipid-soluble, you're going to do it wrong. Okay, here we go. So another thing that differs in our classes of hormones are the pattern of secretion, when the glands or how the glands are releasing that hormone. Okay? What we're going to see is that some hormones, these glands have a chronic secretion, right? So they're releasing that hormone in low amounts, and the hormone stays more or less steady in the blood, right? It has a set point, right? So it fluctuates a little bit around there, but we have more or less this constant release. Thyroid hormone is an example here, right? It's just like always out in the bloodstream. Other hormones, we're going to see this acute secretion. If there's a big stimulus, we have a big release of hormone. If there's a little stimulus, a little release of that hormone, right? But it's quick and it's in response to something. Epinephrine, also known as adrenaline, is your great example here. Episodic hormone secretion, you'll see that this is fairly predictable, right? We have the same size stimulus and there's this repeating pattern. This would be something like some of the reproductive hormones, particularly when we get into the female reproductive tract. There's this rhythm, this cycle of when the hormones are released. And so, for example, this could be luteinizing hormone. Once a month, it's triggering ovulation. And so that is going to be something else that we'll see that varies among hormones. Okay? How do we control that secretion, right? So if these are the patterns of secretion, then the question becomes, okay, well, how do we decide, right, what is causing this? There are three things that are going to trigger hormone secretion, release from that gland. Humoral stimuli, neural stimuli, and hormonal. This gets a little bit confusing to people, but let's take a look at this. So humoral, this is a weird word that people don't use so much anymore. The humors of the body are the fluids, right? So that, like, when they used to like bleed people or say your humor is cold or hot or whatever. It's the fluids of the body, okay? And so all we're saying here is that some changes in levels of different molecules or nutrients can actually trigger the release of hormones. So here they picked a great example, right? So just in the humors, in the blood, if calcium levels start to drop, that actually triggers the release of parathyroid hormone, which tells us to remove some of that calcium from the bone. Remember the osteoclasts, the scrubbing bubbles, sending that calcium out into the bloodstream, and we fix the problem. The opposite works as well. If those calcium levels, right, there's your molecule or your nutrient, it's in the humors, starts getting too high, you don't get parathyroid hormone secretion, and you'll start to drop the levels in the blood, right? So that's the humors. The same thing works with sodium levels. We have a hormone aldosterone that's going to be released in response to that. Even a nutrient, glucose, right? Insulin is released in response to glucose. Okay, so humoral stimuli. Neural stimuli. This kind of goes back to that whole connection with the brain that I was talking about, right? So the big example here is actually the adrenal medulla. So we'll talk about the adrenal glands. They sit right on top of the kidneys, and they're what produce epinephrine, which is adrenaline, right? So this hormone that you need for a fight or flight response, right? You need it quickly. And so what we'll actually see is there's a neuron sitting way up in the hypothalamus in the brain. That axon reaches all the way down to the adrenal medulla, and the neurotransmitter release can actually cause the release of epinephrine into the blood. So that would be neural stimuli. Okay, and then hormonal stimuli. There's a lot going on in this picture, right? But the thing we're going to see is that particularly a lot of the hormones that come from the anterior pituitary gland are what are referred to as tropic hormones. They're actually going to cause the release of other hormones. So they don't have an effect in and of themselves. They're just going to an endocrine gland and telling it to release hormones. So that's your hormonal stimulus there. So kind of a classic example there. The hypothalamus is going to send a releasing hormone to the anterior pituitary, and say we get it releasing follicle stimulating hormone, which then goes down to cells in the ovaries, right, the follicle, and tells it to help an egg develop, right? So that would be an example of a hormonal stimulus. Here's a good one to keep in mind. You will see this a lot from the hypothalamus. When it's trying to control the anterior pituitary, I'll get into this more next week, but here's your hint. It's either sending a releasing hormone and telling the pituitary, okay, release that one. So that's also an example, right, of a hormonal stimulus, the hypothalamus telling the pituitary to tell, right? It can also release inhibiting hormones, which would say, don't release this from the pituitary gland. Anytime you see inhibiting hormone or releasing hormone, it's coming from the hypothalamus. That's your hint for next week. Okay, when we're talking about the endocrine system and how it's set up, it works like pretty much everything in the body, right? We're trying to maintain homeostasis. And so as far as feedback mechanisms go, I will say that almost all of the endocrine system works on negative feedback. So the idea being that as you start, say, you're releasing follicle-stimulating hormone, that cell in the ovary is helping an egg develop, and at the same time it's releasing estrogen, right? And so as there's more and more of that hormone being released, the estrogen actually might have a negative feedback on things like the pituitary or the hypothalamus and saying, oh, we've got enough. No more eggs, we have plenty, right? And so that's negative feedback, shutting it down. There are some examples where you have positive feedback, right? Where we run into this system where the release of that hormone accentuates the release of more hormone. But by and large, negative feedback is the dominant thing here. Okay. We're almost there. Okay. Mechanisms of action. This part of the chapter got kind of crazy. I'm going to ask you to keep coming back to figure 17.9 because it makes the most sense in my mind. Okay? So, the whole point of releasing a hormone is to get a response from that target cell. But what this is showing us is that these things happen slightly differently. Okay. Some hormones use a nuclear receptor. They actually dissolve through the plasma membrane, go directly to the nucleus, and are able to activate genes to create new proteins, and that is the cell's response. Do you think those are lipid-soluble hormones dissolving, diffusing through the cell membrane, or water-soluble hormones? Lipid-soluble, right? Those small polar molecules are actually able to go directly through the phospholipid bilayer of the cell membrane, right? And so lipid-based molecules, lipid-based hormones, lipid-soluble hormones, are going to use nuclear receptors. And we'll look more at what that looks like. The membrane-bound receptors are used then by the water-soluble hormones. And the book kind of kept, I don't know, going in circles around it, but that's really what it comes down to. Okay? Hormones can only affect cells that actually have the correct receptor for that hormone. So for example, what's a good example? Follicle stimulating hormone, right? It's telling the ovaries or the testes to produce more gametes. It's floating through the bloodstream, but it doesn't talk to any other cell in your body because that cell doesn't have the correct receptor. So this is figure 1710 that kind of shows that, right? So this hormone works on this cell, but this one doesn't. Okay, so that's kind of basic there. The key thing to think about with hormones is that there is an extreme level of specificity there, right? It's a lock and key mechanism. The hormone has to fit a receptor in order for it to work. But we've actually utilized this in pharmacology, right? So we've created a lot of synthetic drugs. They can either be agonists or antagonists. So an agonist is actually going to mimic the action of the hormone, so it would actually dock to the receptor and have the same effect. The antagonist kind of drug would actually block the action of the hormone, possibly blocking that receptor so the hormone can't have its action. So you'll see that when you get into, say, like a pharmacology class. Okay, these target cells, this always confuses students, and it's kind of crazy, but these target cells get to change how many receptors they have. So they kind of get to change how much they're listening to these hormones. And so the cells can do two things. This first panel up here is showing downregulation. So there are times when that cell starts actually removing receptors from the surface of the cell, and we see them getting broken up. That would keep you from hearing as much, right, from that hormone that's circulating. So this seems kind of weird. And so some hormones, the example they give in your book I thought was actually really good. So gonadotropin releasing hormone, it comes from the hypothalamus. It's actually released in pulses to prevent the cells in the pituitary from undergoing downregulation, right? For them to say, like, we're not going to listen to you. It's released occasionally, so it doesn't have a chance to do that. There's some look, even like in type 2 diabetes, when you have this resistance to insulin, there's a downregulation of insulin receptors in the surface of the cell, so that might be another example. Upregulation is going to be that when that hormone binds, that one of the responses might actually be to put more receptors out onto the surface of the cell. Again, a great example your textbook uses is follicle-stimulating hormone, and this is interesting. Follicle-stimulating hormone actually causes the production of receptors for luteinizing hormone. So that's going to be really important, particularly when we get to the reproductive system. But realize cells can either pull in some of the receptors or add receptors to the surface. Okay? All right. What we need to do here to wrap things up are look at how these two different types of receptors work. Okay, so here's our lipid-soluble hormone diffusing directly through the phospholipid bilayer and moving into the nucleus of the cell. Okay. The nuclear receptor that's often in the nucleus, occasionally it'll be in the cytoplasm and then go in with the hormone, but realize our activity here is on the nucleus. And what we're going to see happen here is that the hormone is actually going to attach itself to the DNA. So there's a portion on the DNA called a hormone response element. And when the hormone docks to it, this actually becomes a transcription factor, which means you have to think back to Gen Bio, right? So this is going to be a factor that's going to start transcription, meaning we're going to make messenger RNA. And so that messenger RNA then goes to a ribosome and turns into a protein, right? So when you go straight into the nucleus, you can directly affect that gene expression. That sounded complicated until you get to what the water-soluble hormones are doing, and then it gets all sorts of crazy. And we're going to try to simplify it here. The thing to keep in mind, so these membrane-bound receptors are, of course, in the membrane. Here we see the hormone docking to it. There's a few different things that these receptors might do. Sorry, that is the lipid phase. Okay, here we go. So one of the things that very often happens is it's triggering off of these membrane-bound receptors. It triggers what's called a G protein. This is actually a little series of proteins, protein subunits that are embedded on the inside of the membrane. And these G proteins then can do a bunch of different things. So we've activated the G protein, and that might do something like trigger another enzyme. So these, I pulled all of these out of the book. There's adenylate cyclase, right, which then triggers the release of cyclic AMP, which triggers this protein kinase, another enzyme. So the way I want you to think about this to simplify it, right, is you should know that these G proteins are often triggered due to the membrane-bound receptors. you should recognize G protein as part of this because it's a common target, again, for pharmaceuticals. But these G proteins then can do things like activate enzymes, as we saw up here. They might open ion channels. Okay. There are some receptors that don't use the G protein that we see taking place here, but again, we're activating enzymes, or in this case, we're phosphorylating. Phosphorylation is just a fancy way to say you're adding phosphate groups onto something, but that's a really common way in the body to activate things. Okay, so this gets a little crazy, right? But if you take it back to nuclear receptors versus membrane-bound receptors, let's look at what we got here. Nuclear receptors activate genes, synthesize proteins, that's the cell's response. Here's our membrane-bound receptors. Some of them alter G proteins that can open or close ion channels and activate enzymes. Other receptors might activate enzymes and phosphorylate in a down, right? This is what you really want to know. Okay? Oh, that was the other thing I was going to say. Think about this. Membrane-bound receptors say they're activating enzymes. Those already exist, they just have to flip them on. All of your lipid-based proteins, right, if you're actually going through this process of transcription translation, not that it's slow, but this is slower, right? And so this is going to be more gradual, long-lasting things. These can have some quicker effects. Okay, very last thing. One of the things to realize is that through particularly the membrane-bound receptors, we get this process that's called amplification. So if a hormone binds to a receptor and a couple, they're showing 3 G proteins getting activated. But then that activates, we have there 10 enzymes, right, cyclase enzyme, which then creates, 21 cyclic AMP molecules, which then activate this other enzyme, right? We get, 40 of them. This is called amplification. And this is a really important way that cells can take minute amounts of hormones and actually get this very large effect out of that hormone. Okay. And again, that is with those membrane-bound receptors. You'll often hear some of these processes, for example, here referred to as a second messenger system, right? It's like the hormone bound at the receptor, a G protein is activated, cyclic AMP is your second messenger, right? It's like this relay of information. But one of the important things about that is that it can really amplify the cell's response. Okay, we're going to leave it there, but I do want to remind you, on Thursday, when we meet in WebEx, I already mentioned we're going to talk about, I'm going to have you guys go through and kind of do a compare and contrast of the nervous system and the endocrine system. So read through the chapter, get yourself prepared for that. Look back at the nervous system if you need to remind yourself, if AMP1 has been a while. And then the other thing that I'll have us do is maybe a little compare and contrast of lipid-based hormones, lipid-soluble hormones versus water-soluble hormones. Again, to kind of try to synthesize and organize our thoughts around the differences between those two types of hormones. So spend a little bit of time maybe looking at that, so when you're talking with the group, you can contribute. Okay, we'll see you on Thursday.